Good afternoon, everyone. My name is Antonio Gonçalves de Silva, and I am the Executive Vice President of the European Federation of Chemical Engineering, EFCE. I've taken on the role of Executive Vice President in January 2026, following one term as member of the Executive Board of EFCE. I am a chemical engineer by training, graduated from Instituto Superior Técnico, the engineering college of the University of Lisbon, Portugal. I had an industrial career in the areas of project, erection, commissioning, and startup of industrial units. And in industry, I also worked in product development, customer service, logistics, quality, and business management. At the same time, I had an academic career. For more than 30 years, I was a professor of chemical engineering at the Lisbon Politecnico. Finally, in the last 19 years, I had several positions at the Ordem dos Engenheiros of Portugal, the Engineers Professional Association, and lawful regulator of the engineering profession in this country. Two simple conclusions about myself. First, I'm very old. Second, I've been very busy. I'm very happy and honored to be involved in the introduction of this webinar on crystallization for pharmaceutical applications, and I'm looking forward to participating in it. Let me say something about the Spotlight Talk series now. This is now our 6th year of the Spotlight Talk initiative, which started in 2020 to keep together the community during the COVID pandemic. The success of the previous years convinced us to transform the Spotlight Talk into a regular feature of the EFCE program. We have now one Spotlight Talk series in odd years and two in even years, such as 2026. This spring, 11 of our working parties and sections, namely mixing, multi-phase fluid flow, membrane engineering, static electricity in industry, crystallization, drying, chemical reaction engineering, process intensification, education, thermodynamics and transport properties, and early career chemical engineers are delivering over 2 weeks, 9 webinars of 3 or 4 talks focused on specific topics by leading industrial and academic experts. The series also enables attendees to sample matters in new areas, encouraging cross-fertilization of interests. The complete program is available on the EFCE website. EFCE promotes scientific collaboration and supports the work of chemical engineers in 30 European countries, representing more than 100,000 professionals in Europe. EFCE's working parties and sections cover all major specific aspects of chemical engineering and are in fact at the core of the organization, forming the scientific engine that drives many of EFCE's activities. They provide an important forum for technical exchange and networking among chemical engineers in Europe. Before concluding, I would like to thank all the colleagues who worked hard within the working parties, sections, and wider EFCE for this initiative to happen and to be successful. In particular, many thanks to EFCE's Martin Pou in Toulouse, who did most of the conceptual setting up and practical activities. Merci beaucoup, Martin. I thank you for your attention, and I wish all the speakers and the attendees a fruitful and successful webinar. I would now give back the word to the chair of this event, Botond Szilagyi, to start the webinar. Thank you all for your attention.
Thank you very much. Thank you, Antonio, for this very nice introduction and opening this Spotlight series, Spotlight on Crystallization. Thank you for giving an overview of the Spotlight talks in the EFCE. And thank you, Martin, for organizing this talk so smoothly and nicely. Let me introduce myself. My name is Botond Szilagyi, and I'm an associate professor at Budapest University of Technology and Economics. And unfortunately, Daniele Marchesi, the chair of the crystallization team, who I present today. So Antonello, Raponi, and myself will moderate this Spotlight talk today. Let me share my screen and transmit the message of Daniele Marchesi on the crystallization PEP. Can you please confirm that you can see my screen now?
Yes.
Yes, thank you. Before jumping into the details, let me give you 3 technical announcements. The first announcement is that the attendees of this webinar will not be able to speak. The microphones are muted. If you will have any questions to the presenters, please use the Q&A session of this Zoom meeting. Type your questions there. And after the presentations, we will read your questions and the presenter will be able to react. If there will be too many questions, then the presenter can respond for you in the chat window. The second announcement is that this webinar is recorded and will be displayed on YouTube. The third announcement is, unfortunately, one of the speakers, Zoltán, could not make it today to join because of traveling complications, which means that we will go ahead with the 3 planned presentations and we can leave a bit longer Q&A and discussion time today. All right, with all those technical announcements being said, let me present the working party on crystallization of the European Federation of Chemical Engineering in Danilo. What is the structure of the VP? The VP is made of 2 delegate members for Society of the European Federation of Chemical Engineering. And each member society delegates 2 members. Typically, one of them is an industrial and the other is an academic member of the VP. At the moment, there are 22 members from Europe, and it's important that the mandate of these members will expire soon, so there will be another review of the crystallization at least. Beyond these delegated members, there is a varying number of elected and quest members, not just from Europe, but from all around the world, to represent a mix of scientists from all around the world of crystallization science. The aim of the crystallization VP is mainly to organize events, foster collaboration, and to promote interdisciplinary research. Of course, the crystallization VP does this around the crystallization. The crystallization VP covers fundamentals of crystallization, theoretical and experimental methods of analysis of crystallization processes. And it also includes application of crystallization in different sectors, from pharma to food, agrochemicals, and now more recently also in battery applications. Of course, there is always an emerging applied research line of crystallization also from the precipitation of biomolecules, the battery materials, critical raw materials, and of course these are also being tracked and taken care of by the crystallization VP. One of the main events organized by the VP is the International Symposium on Industrial Crystallization, and the last edition was organized in Glasgow, Scotland, in 2023, together with the annual meeting of the British Association of Crystallography, BACG. The VP also organizes thematic sessions in the European Congress on Chemical Engineering. In the last edition, the session was entitled "The Pharma Challenge: A Cross-Cutting Perspective" in 2023 in Berlin. And of course, the VP organizes webinars like this, and summer schools and other events. Something to highlight was a summer school workshop on small molecule crystallization, which was organized in Torino 2 years ago, which was a very nice and successful workshop on small molecule crystallization. And there are also summer schools organized mainly for students and everyone who wants to hear and learn about crystallization, usually in every other year. I would like to turn to one of the flagship crystallization events around the globe, which is being organized right now by the EFCE crystallization VP, which is the International Symposium on Industrial Crystallization, to be organized in this year from 6 to 9 September in Budapest, Hungary, in the hotel area. If you are not registered yet, you are invited to join, because this is a 3-annual conference, which means that the next edition will be organized in 2029. Yes, you can still register. You are still within the early bird deadline, and you can still bring your own abstract to be presented as a poster at the conference. Usually, there is a mix of industrial and academic attendees, which fuels very fruitful discussions. We have a good lineup of plenary speakers already announced, and one of the speakers that we will have today, Neda Nazemifard, will also be a plenary speaker in the IC conference to be organized in Budapest. There will be an EFCE crystallization award presented at the ISIC conference. And I displayed here the last three winners of this prestigious award, Francesca Bragaglieri, Ashwin Rajagopalan, and Elena Simone. The next one will be announced today, and you are still on time to nominate your talented PhD student who graduated recently. The award winner will not only be invited to the conference free of charge, but a travel grant will also be provided, and it will be given the possibility to present the thesis as a plenary lecture. So everybody will listen to that student. I hope to see you in Budapest in the ISIC, and with that, I would like to hand over the introduction to my fellow Antonello Raponi. Antonello, floor is yours.
All right. Hello, everyone. It's so nice to see you all here. My name is Antonello Raponi. I was a postdoc fellow in Purdue University and recently joined Ecole des Mines Saint-Etienne as associate professor. And I will now share the screen to briefly introduce you some initiatives done in collaboration with Professor Ottman, University of Lyon. Can you please confirm you can see the screen?
Yes.
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 All right. Which are the population balance modeling webinar series and population balance modeling conference roadmap. So this is just a short introduction for our growing community. I'd like to shed a bit of light on the third edition of PBM webinar, which is a low barrier format that keeps the field active between large in-person milestones, including somehow this one. It keeps researchers connected across universities, domain, and continents, gives visibility to early career work and fresh results, brings together formulation, numerics, experiments, multi-scale coupling, and applications, and in the end lets new ideas mature before they reach the conference stage. In short, population balance modeling webinar is not only the next webinar, it is what bridges today's conversation to the next PBM conference. So what was done? It was the population balance modeling webinar 2 in 2025 and shifted to 2025, where we got nice entertainment among several PhD students and postdocs. And it was a one full day with morning and afternoon sections, and it was especially useful because it allowed us to discuss on relevant new topics, such as the AI integration, which was back then very hyped. And so this brought a very strong, for example, participation in terms of collaboration between different universities, including Politecnico di Torino, Purdue University, and other one in Europe. So why is that important? As said, it offers a low barrier format and creates space for exchange across a very broad range of application and topics. And these are just some you can see that were presented during last webinar. At the same time, we are also looking ahead to PBM conference, which will be PBM 2028, which we are starting to organize again in collaboration with Politecnico di Torino, and the lead will be taken by Professor Marchisio. Our goal is to make it a strong meeting point for the entire community, bringing together methodology and methodology advances, industrial application, and emerging directions. So population balance modeling webinar and PBM 2020 are really part of the same roadmap, as you can see here. One keeps the momentum alive right now, and the other one will be the next major place where the community meets in person. So with that, I would like to invite you all, of course, during this next meeting in Torino, and looking forward to meeting you there. So with this, let me switch to the other presentation. All right, all right. All right, so these were the initially four planned speakers, but unfortunately today Professor Naj won't be able to join us. And so this is the real kickoff of the crystallization for pharmaceutical applications, where the speakers will be Dr. Neda Nazemifard, Dr. Lorena Pasero, and Professor Christopher Burcham. So, all right. So I would start with Dr. Neda Nazemifard from Takeda, United States, who will present and deliver a presentation named In-silico-first strategy for designing crystallization processes for pharmaceutical APIs. So a brief introduction for Neda. She is currently a fellow and process modeling lead in small molecules process chemistry development at Takeda. And she leads process modeling activities across the process life cycle, from early development to risk assessment and validation. And previously, she was associate professor at University of Alberta, where she led research on complex fluids in micro and nanofluidic platforms. So, Neda, thank you for joining us, and I leave the floor to you.
Okay, thank you so much, Antonello. Can everybody hear me?
We can.
Awesome. So let me share my screen. Okay. Okay. Can everybody see the screen as well?
Yes.
Awesome.
Yes.
Okay, thank you so much, Antonello, for the kind introduction, and thanks for the invitation from the European Federation of Chemical Engineering. My name is Neda Nazemifard. I'm a fellow at Takeda Pharmaceutical, and today discussion I would like to I'm excited to share our plan on in-silico-first strategy for designing crystallization process for pharmaceutical APIs. So essentially, this talk is about how we are digitalizing and modernizing crystallization process development using modeling, automation, and data. What I want to share today is basically how we're trying to make crystallization development more in the context of in silico first. So it basically does not mean replacing experiments by models, but it means using experiments differently to generating better data, connecting those data to models much earlier, and then using the models that we still, in a time that we still have opportunity to change the process or impact the process, basically. So over the next basically 20 minutes or so, I'll walk you through case studies, our approach, and how we made an impact basically into our projects. Okay, so this slide basically kind of put the vision behind the in silico first. So one important point that we are trying to make using this schematic is that digitalization or in silico first is not one model sitting on one person or scientist, basically laptop. In crystallization, it has to connect the full stack, as you can see. As the bottom layer, this is where we have automated and data-rich experimentations, high-throughput screenings, PAT-enabled reactors and crystallizers, and data-rich experimentation and stations. These data only becomes useful when we capture the full context in a structured way. So having fair data bases that captures materials, properties, vessels, process context, and reaction database as well. The next level that has to be connected to this data is our models. So thermodynamic models, kinetic models, population balance models, CFD are optimization tools that then work with these data. And then the last step, which is probably one of the most critical part, the output has to come back to project team through simple interfaces, web apps, or AI agents. So it can influence different decisions, solvent selection, for example, in crystallization, pathway design, scale up, and control. So the real goal here is a closed learning loop, basically, between all of these layers, between data, model, and decision. To our basically perspective, digitalization only work when the layers talk to each other and they are connected. So to us, the architecture is as important as the science and the technical side of each of these elements as well. Now, this is sort of one of the areas that we prioritized as synthetic molecule process development team was to basically apply the in-silico-first perspective on crystallization as one of the main unit operations that it exists in all of our unit operations. So this is the roadmap that I basically talk you through, and this is what we laid out for the crystallization process design. So the first bucket, if I want to break the work into 4 modules, the first one is crystallization pathway selection, basically. So this is mainly thermodynamic, choosing solvent system, crystallization mode, and understanding impurity behavior. The second one, or the second bucket, is where we bring in kinetics, and we are looking at nucleation, growth. Basically, we involve population balance models to optimize and design the crystallization, basically. The third layer or bucket is development of digital twins, and here where we are really looking at the scale-dependent and hydrodynamic effects. This is where these impacts start to matter much more. And the last bucket is basically the crystal, the fourth one is crystal property prediction, and that can be the morphology, form, basically polymorphism of these crystals. So the important point is that they are not 4 separate modeling exercises. They all sit on a common layer. It's a data backbone where we are really committing into a fair data capture. So every experimental data goes into the structured database to fuel basically our machine learning AI models. So solubility, for instance, all the measurements with all the context would go there in order to build machine learning models for our solubility, basically. And then the data would be captured at each step. So here is basically module 1, sort of pathway selection. So this is, again, it's in early development. We never have perfect data or a lot of samples to measure a lot of solubility. So that is exactly why we need a staged workflow. So we just start with basically a data-free or very low data model to rank the solvents and identify promising solvent-antisolvent pairs. Then we run a focused high throughput screening, not a broad empirical sweep, and this gives us enough information to refine the model and generate some yield maps, basically. After that, we invest in more detailed solubility and metastable zone work only for the down-selected systems. So instead of measuring everything everywhere, we sequence basically the learning. And every measurement, every experiment would be used for refining and having a better model for solubility predictions. So here is basically, this slide shows the thermodynamic starting point. So crystallization design is really anchored in solubility, supersaturation, and metastable zone. So if this foundation is solid, we can really have a very robust crystallization design from the beginning. So the question is that we are asking is that, can we design a viable pathway from minimal experimental data for a new API if I know the structure and few key properties, basically, such as a melting point and enthalpy of the fusion, I can already basically design predicting solubility behavior, screening solvent, estimating yield, and looking at slurry density. So then I use a limited amount of measured data to connect or calibrate the model, as I said before. So the point is not to believe the model blindly, but the point is to use it early enough to avoid locking into solvent system or operating window that is fundamentally working against the outcome, basically, that you want. And that is especially, I think, is very valuable in pharma when material is very limited and scarce, and then the cost of an early, basically wrong selection is high. So it also helps us to ask practical question early on. So do we really want cooling, antisolvent, or hybrid route for our crystallization? And this is likely through a throughput limit. So we can put constraint on our throughput, on our slurry concentration, and basically our yield as well. This slide shows that basically, so one thing we learned very early was that these models only scale if the project team can actually use them in everyday, basically, development. This is why we develop a crystallization process design web application, basically, and this workflow was developed in collaboration with MIT through the postdoc that we do have. So this slide is basically a snapshot of what that app interface would look like. So the idea is to guide the scientist through the In-silico-first workflow, basically via a software tool. So for example, you enter the API properties like a molecular structure or key properties, and then choose what sort of input data you have. So if you have no data, or if you have very limited, or you can basically use start from COSMO-RS or an extended Hansen method or UniFact, dependent on the situation and amount of data that you have. You can then basically move on into looking at cooling, antisolvent, hybrid, or all of those basically mentioned scenarios, and indicate that you want to account for basically calculations of your purity, of your yield and solvent concentration. And you can put constraint here as we show here, and by using this constraint based on the measurement and the model, you can basically pick some of the choices that would satisfy and basically be within that constraint or window that you put here. So that the workflow, it will be developed more into considering some of the thermodynamic part of the impurity partition coefficient calculation as well, starting from maybe simple differences between the main compound and solubility, the difference between the solubility of the main compound and the impurities, maybe starting with no data models, because usually when at this stage we don't have the impurities being manufactured or synthesized. So we start with no data model, and that relative impurity or compound solubility can also give us a lot of information during the solvent selection as well. But this is the part that is going to be developed to be more mature and incorporate other ways that impurity can be entrapped within the structure, in addition to just simply solubility differences. So, so this is kind of an example that we wanted to share. So this is an example from a real compound, compound A. So we started from COSMO-RS predictions from the structure and fusion properties, and then add a limited number of experimental measurements. That hybrid approach basically let us predict solubility in solvent systems that we never really directly measured. Once we have that, we can rank the solvents, basically estimate the yield, and also look at the slurry density, which matters a lot for practical processability. Here we can also look at the classes, the solvent system classes. We can bring in cost, we can bring in sustainability factors. We can filter these pairs by miscibility, so it can really provide a strong tool for our scientists and engineers in order to narrow down the solvent system based on different metrics. So as you can see there, this really acts as a filter. So then the wet lab crystallization work is focused on the most relevant system. So in other words, the model, as I said, it's been really acting as a filter. So it would tell us what measurement or what solvents have been down selected by just solubility. And it would focus our experimental effort when the value is really highest instead of treating every solvent system equally. So for early development, again, this is exactly the kind of leverage that we are hoping the model can provide. So for instance, for this case, we saw a very basically favorable system with the MSO water. We also narrowed down a couple of other systems based on just solubility and yield and concentration of the slurries here. Another example that we want to share here is basically, this is not part of, so here we were looking really at solubility workflow and solubility screening. So specifically trying to find a more greener solvent alternative for an API that was previously processed with a not so friendly solvent, as you can see basically. So we had to choose a solvent system for spray drying of a compound, and existing manual solubility screening didn't really yield any promising hits. So the traditional approach was used to manually look at some of these experiments in the lab, and we didn't find a really good solvent from a typical solvent that we usually test. We also look at mixture of solvents, and they were manually measured with solubility, but again, no suitable system was basically found. So here is really where we went through our workflow. So starting with no data, Cosmo RS was used to predict solubility in unexplored solvents. So we looked at 185 solvents in a very short time. And then the solubility model suggested the formic acid, acetic acid, and pyridine and allyl alcohol as some of the good candidates. And these are not some of the typical solvents that we usually look at. So then out of this, we went to the lab, we run some measurements in these solvents in some binary mixture. We bring back this data basically to the model, we refine the model, and out of that, we basically had a ternary system of formic acid, anisole, and methanol that was eventually selected basically out of these a loop of, as you can see, the in silico and automated HTE and bespoke experiments that was chosen, and this is what the project team are basically using. So this whole activity take probably a few days to a week, and we managed to find something that is not really part of the typical solvent system that we would really use. So this is probably perhaps a common problem. So earlier screens are often shaped by what people typically try, not by a full chemical space. So this was a good situation to ask whether an in silico string really could push us beyond the obvious choices and expand the search space without exploding experimental workload. So I think this is where these digital methods are most useful, not when they confirm what we already know, but where they actually help us to ask better questions and consider a bigger space of our parameters. So this one is basically, so if we're basically moving from that first module, which is just focused on thermodynamic solvent selection and pathway. So here, once we have a thermodynamically feasible solvent system from an in silico screen, then we need to focus on kinetic evolution workflow. So on those shortlisted candidates, that typically involves several type of experiments that we would run, and we would like to understand how is the crystal growth or nucleation would perform within these systems that we down selected from solubility workflow. So there are a couple of experiments that that, it's been described in literature as well. So polythermal experiments like cooling curves, basically, for example, using a crystal 16 unit to see the crystallization and dissolution points, and basically isothermal hold experiments using something like crystalline setup to monitor how crystal nucleates or grow at constant supersaturation. Desupersaturation tests basically to look at nucleation and growth rate, and even stress testing in small reactors like EasyMax to see how robust a system is under different conditions. So by doing this, we qualitatively or semi-quantitatively rank the candidates from a kinetic perspective. So basically nucleation causing many fine particles or etc. This let us make an informed down selection so we can pick the best solvent system and crystallization mode for further development. So again, because in the earlier development, we may not know the exact critical quality attributes or CQA that we have in our process. So we use these experiments to map out an attainable region. And this is something that has been published basically, and the terminology that been used and published before. And we are really trying to get to this region by early smaller scale screening basically, and automation and modeling, so we can have that region that would provide us a lot of flexibility moving forward, so we don't lock ourselves in solvent systems that are not favorable to our CKAs in the future. So here, again, you can see that for the same compound A that I showed you, we use the solubility basically screening or in silico screening for narrowing down in a couple of solvent systems. We are looking at the MSO water. One was that selected from the solubility, and the other one which showed a good yield and slurry concentration was THF heptane, for instance. So we run these experiments. We are looking at purity, yield, form, basically fouling tendency, agglomeration tendency, and some kinetic in measurements are basically ongoing as well. So this gave us a very good tool in order to then select the system that we want to go and really focus on optimization of the crystallization. So this part really, this is the third. So this is where we are really looking and bringing all of those measurements of the kinetic into building a more basically direct models to assess and optimize crystallization. So once the route is down selected, this is the modeling framework we use for optimization of the crystallization. So at the center is basically a population balance model with the mechanisms we expect to matter. So primary and secondary denucleation, growth, agglomeration, breakage. Hydrodynamic also has to come in the picture here because collision environment, shear, and mixing can influence several of these kernels, as we know. So we then use optimal experimental design to choose a small number of runs that be truly informative, and it basically help us with both parameter estimation and model discrimination. So the validated model then can go to several jobs for us. It can generate design maps, predict risk, and support the normal operating regions and proven acceptable ranges, and suggested scale-up recipes for different crystallization mode. This is basically the workflow that we developed in collaboration with Purdue, following their IMED workflow that uses the iterative method of model-based design for designing best experiments, most informative experiments that they can be used for model discrimination, and also the basically parameter fitting. So this is basically case study. So now let me give you basically one of the case studies that we developed, and we use this IMED approach in order to develop the population balance model here. So we wanted not just to model crystallization, but we also, so we wanted to look at this model and use this model for different objectives in the project. So perform a sensitivity analysis to understand which parameter most strongly affect the outcome. We used it to inform a preliminary risk assessment, basically identifying where we could go wrong and what we should control tightly in this crystallization, and identifying key process parameters that we would need to focus. For example, maybe the antisolvent addition rate or the basically cooling rate would be some of the most important factors impacting the crystallization. So here we used a couple of basically data-rich experimentation in the beginning, around 5 and 6, to start the building of the population balance model. Then as we go through the workflow that I showed before during the basically the model-based design of experiment, we gradually added more experiment. These are all very data-rich experiments where we measure the concentration. We had basically looking at a lot of different orthogonal methods for calculation of concentration and size, and how it would change during the experiment. And using these number of experiments, basically we managed to build a population balance model for the crystallization, as you can see. Now, as I said, we use this model to help us with a lot of understanding of this crystallization, basically. So as you can see from here, so we managed to do a full experimental factorial design, in silico factorial design with a fraction of the time and basically experimental time and material that would have been taken to do the same amount experimentally. And the model identified two key process parameters that really mattered for controlling the particle size. So antisolvent feeding rate and supersaturation level during seeding were critical levers that we managed to understand through this in silico DOE. So things like, for example, holding time and cooling rate had negligible impact on the particle size distributions. So by using the model, we could really focus on what mattered most very early in the process. In addition, the model prediction provided data to support the risk assessment for the crystallization. So traditionally, to thoroughly and assess the risk, like what happens if we deviate from the ideal, for example, feeding rate, you have to run a bunch of extra experiments, but here we can really simulate these scenarios. So it also showed us to explore more complex what-if systems that, for example, if we have a nonlinear dosing profile, how that might impact or test the crystallization without doing it experimentally. So in summary, the PBM model combined with this in silico DOE let us do in days basically what might have taken weeks otherwise, and it also pinpointed the key factors for a robust process. The next module is basically development of digital twins. And as I said, this is basically where we are really bringing the hydrodynamics into the picture, and we are looking at the scale impact. So this is also done with collaboration with Purdue. So this is really where we touch on, as we touched on this earlier, but this is where taking our models to the next level by ensuring that they represent the real world process, equipment and scale. So a digital twin of a crystallization process means that we have the simulation model that runs in parallel to the actual process, and we are really capturing the impact of the scale, how that would impact our nucleation, our basically agglomeration, our basically kinetic of the growth, and really bringing that into the picture so we can use the models to give us recipes for scale up and also for really robust scale up, and also design the experiment for the manufacturing and demo batches based on these model predictions. So we always stay within the range of our acceptable range of our basically CQAs. So in order to facilitate this type of activities, we noticed that one of the basically our bottlenecks are having these CFD simulations ready to use when we really need them, considering the very tight project time. So this is an effort that also started in order to facilitate this building of the digital twins. So building our vessel library, basically, which include all of our internal reactors, all of the basically external entities and CRO, CMOs that we work with, and building a comprehensive vessel library with accurate basically geometry and a specification of these reactors and crystallizers, and then start to build CFD models in the back ends that we can really use later on with surrogate models or reduced models in order to get a very good approximation of macromixing time, power per volume, and shear. So we are using a suite of different type of modeling. CFD, of course, is the most accurate, but it's the one that probably takes the most amount of time and computational cost. Software such as DynoChems we are using basically, but we know that there are basically empirical equations that are very specific for standard geometries. So the accuracy might be lower, but they are probably in terms of cost, they are lower than the CFD simulations. And then what we are building is probably yield in between. So we are trying to develop many CFD models for our reactors and crystallizers. And as I said, use this to build reduced models that are developed for that specific geometry. So here basically shows a couple of, what we are intended to do with these ones. So starting from lab scale simulations, going to pilot and basically manufacturing in order to use all of these hydrodynamic properties for the scale up of our crystallization, basically. The last bucket that we have here, so here we are really using the in-silico-first crystal property predictions. So here can be the predictions of form, morphology, and other particle properties. So in pharma, we know that CSP is used to predict possible polymorph of a molecule at different crystal form and that can take. But however, these predictions are theoretical and need to be vetted. So in addition to basically experimental screening that every basically company do, this is also an in silico workflow listed here is above that we can access, we can assess really this crystal structure prediction result critically. So we do DFT or density functional theory to calculate the minimize the energy and optimize the geometry of the predicted crystal structure. This helps us to check if the predicted structure is truly a stable minimum or not. We calculate free energies, including 0-point energy and thermal basically corrections to compare relative stabilities of these forms. We perform hydrogen-based analysis basically and packing similarity analysis using tools like CCD, basically mercury, to see how the predicted hydrogen bond networks are there any bizarre behaviors going on or not. We also do computational basically analysis generating conformers via molecular dynamic or other methods to see if the molecule can actually adopt the conformation that the predicted crystal structure would require. So these are basically some of the tools, in silico tools that we are using to develop in order to assess some of these predictions in addition to experimental screenings that we do. So overall, to wrap up, I kind of wanted to highlight a few key takeaways and future directions. So first, we seen that by embodying the in silico tools and automation into everyday process development, we can fundamentally change how we work. We move away from treating modeling as a separate, exercise and instead use them for real-time decision making in the project. It's basically basically a shift that chemists and engineers are now collaborating with data scientists, automation experts, and the modelers. So second, the in-silico-first strategy let us design processes with end in mind. So we focus on the desired outputs, for example, for this crystallization with high yield, desired crystal form, and maybe focusing on more growth dominant crystallization system from the very start. So this prevents these basically early lock-ins decisions, like choosing a solvent becomes basically it is kind of a work in one early experiment. So we can really use these tools to narrow down or down select our systems the way that would be favorable for the outcome that we would want. So we also stay flexible and data driven until we find basically the best solution. We are also in a regulatory industry, so we know that the models and automation has to meet a high bar. So we're investigating and investing a lot of validation and risk assessment for our models, ensuring that they are accurate, which they're according to their intended use. And basically, we have a very well documented everything on model versioning, basically, and data that has been used for model development and validation. So we also work very closely with our regulatory, CMC, and also quality teams, so that when we eventually take these models into later stages of process characterization and perhaps filing, we are all aligned on the objective. Finally, looking ahead, I think the big focus is on data. So all of these machine learning AI models we talk about can only be as good as the data that they have been used to train them. And we are building these fair data structures and standardizing how we collect and measure some of the things like solubility in order to have a very reliable, fair data structures that we can really use for machine learning. So in summary, In-silico-first is already improving our basically process development activities, and with continued work on validation and data, it's really poised to become an integral part of our process development in order to develop drugs faster and using more robust and sustainable basically processes. So again, to summarize, the value of an in-silico-first strategy, not just one specific model, it is basically the contribution of a staged experiment, data, and fit-for-purpose model embedded in real development workflows. So in this effort has been a very basically a diverse contribution from different teams here that some of them I would like to acknowledge. So many people who have contributed into this whole workflow. So from the SMPD leadership team, from Charles, who is the head of process engineering and technology, and Fred, who is the global head of SMPD. From my team In-silico, basically Baron, who is the postdoc we have with MIT, and she really focused on building our in-silico solvent screening workflow. Yash, as basically who was a PhD student in Professor Naj group and later on joined Takeda as a basically engineer in process modeling team, who contributed significantly and is building our kinetic part of the workflow, from automation, robotics, advanced processing, where they basically help us with screening and as well as development of data-rich experimentation. And from processing engineering team, we have Varun and Suyong, who are part of process engineering and help significantly with building and reviewing this workflow, and Gonc, who is also a senior science engineer in our process engineering team, in basically helping us with development of this workflow. We also, as I said, we work with MIT with Professor Alan Meyerson and Richard Bratt's group, and also Purdue with Professor Naj's group in development of different parts of these workflow. So in finishing this basically presentation, I would like to thank you for your attention. I hope this talk give you a clear picture of how in-silico-first strategy can accelerate and improve crystallization process design. I'd be happy to take any questions or discuss further any of these modules that we discussed.
Thank you, Neda, so much for this great presentation and overview on the in-silico-first strategy in the pharma. I really like that you mentioned that everything has to be integrated, right? So it's not enough to consider just the crystallization. You need to plug in the data level, you need to plug in the project teams, and you also need to consider from the thermodynamics to the process and mixing level, everything for this to work well. We have some questions in the Q&A area. One attendee is asking that in slide 2, how you decide the experiments on the HTE? Is it random sampling in the parameter range, or is there a model to define the next experiment?
So that's what we are trying to really avoid, so we don't do random screening. So as I said, the paradigm, I would say probably this slide shows the best of what is our philosophy in designing experiments for the high throughput screening. Is really to start from models. So if we have, and as much as possible. So this can be coming from, large language model looking at literature, or it can come from our thermodynamic or mechanistic models that need least amount of data. So for the solubility specifically, what we are doing is that we use COSMO-RS, which doesn't need any data. You probably just need to measure some heat of fusion or some, with the structure of your molecule, you can go, you get some hits, and that's where we are really suggesting then some measurements through our high throughput screening of the solubility. So we rely on our basically thermodynamic suggestions that what are the best solvent or what are the most poor solvent that can be used as an anti-solvent. We also just to again, that's the suggestion from Cosmo. But in order to feed the data to maybe a different model, such as the UNIFAC models in DynoChem that needs some data, we also push or force some other solvents that is not maybe recommended by Cosmo in order to cover the whole different class of solvents. So again, this experiments in high throughput, for example, comes from modeling in mind. What are the solvent systems or classes that are missing, and how that data can help us with the next step of model refinement. So it's kind of model-driven or model-informed experiments.
Thank you. More questions just started to appear in the Q&A, so I will ask you, Neda, to respond these during the next talk maybe. But there is a group of questions related to the PBM and the CFD model. I don't want to quote, I will ask you to answer, but basically they are asking about the reliability of the PBM and how complex these systems were that you apply the PBM and the CFD for. Basically, your question is that what if the crystallization, the crystal growth and nucleation mechanism changes during the experiment, right? In different supersaturation, you may have a different growth mechanism which should be modeled with different equations. That one class, and the other is that how do you know if your model is still valid within your design space and you don't need some adaptations, either mixing or mechanistic wise?
Yeah, so I think that's a great basically question. I mean, population balance models are probably one of the hardest, and we have a lot of experts in this community who can probably attest to that better than me. So what we are trying to basically do, that's why I said we are really, we always keep this loop of validating basically the models across different scales. So in the beginning, like experiments that I showed you that we used for the models, they were mostly developed at a smaller scale experiments, maybe 100 mil, for example, or 1 liter reactors. So we try to go through this loop and develop this population balance model for that cases. Then we will bring in the hydrodynamic. So we would run a few experiments, maybe at the different scales, at the CFD calculations, and then then we bring in the hydrodynamic, how that would impact the, as you mentioned, these hydrodynamics would come into our kernels, like how they would impact the growth, how they would impact nucleation, and then we will capture them during these scale-up activities, and then we always have to, again, validate the model. So we usually try to train the model for example, if we use it at 3 liter scale, we use that data to train it, but we always try to validate the model in a scale that the model hasn't seen. So that's kind of one way for us to make sure that what we have captured is correct, and it really translates into different scales as well. But it's really, it's a continuous loop of, as we get more data across the scale, we bring it into our model training and validation, basically. And there's also a case of validation or model accuracy depends on the context of use. So no model is always perfect, basically. So we have to define a criteria depending on where this model is being used. And sometimes you can still use a model even if it has, some uncertainty around it, as long as you are aware of those uncertainty, and also the context of use would allow you to use that basically a model with that level of uncertainty. So this is something that I was trying to kind of emphasize at the end, that if these models are going to be used at later stages of the project and being really used for decision making, we need to have a very good understanding of our uncertainty analysis and validation.
Thank you very much. And I think that answer made a lot of sense and also clarified the role of the PVMs. I think we will need to proceed with the next presentation. So thank you again for this great presentation and overview. And please, Neda, if you can, please stay with us and answer the questions
Sure.
more concretely in the chat area. Thank you so much.
Thank you so much. Thank you, everyone. Thanks.
All right. Thank you, Neda, once again, especially for all of this very interesting insight for bringing up scale up using CFD. So now I would move, let me just briefly share again the screen. All right, we have the second speaker of this of today's seminar, who's Dr. Lorena Pasero. She's from Politecnico di Torino, DSAT, Italy, and she will deliver a talk entitled Advancing Convection-Free Protein Crystallization, Modulating Silica-Gel Interactions to Enhance Nucleation. A brief background for Lorena as well. She recently completed her PhD in chemical engineering at the Politecnico di Torino with a dissertation on particle engineering via spray freeze drying for pulmonary drug delivery application, and her recent work spans inhalable formulation, SFT, and protein crystallization. So Lorena, thank you for joining our seminar, and please, the floor is yours.
Hello, thank you. Okay, I'm trying to share my screen. Okay.
Okay, yes, we see the screen, but not the presentation mode.
Now, can you see it?
Now we see it, yes. Thank you.
Okay, great. So I'll start then. So good morning, everyone, and thank you for the opportunity to be here today and for the presentation. So today I will discuss how we can advance convection-free protein crystallization by modulating silica-gel interactions to enhance nucleation control. This work builds upon the need to reconcile two aspects, which are the advantages of silica gels in convection-free crystallization and also their intrinsic inhibition effect on protein nucleation. And our goal was fundamental and simple. So can we turn down the inhibition effect of silica gels without losing their structural advantages? And so to do that, we should start, let's say, from a brief introduction on protein crystallization, which is employed to determine the three-dimensional structure of proteins, but also to purify and administer drugs. Despite this application and the main advantages, this is a tricky process which commonly relies on a trial and error approach, since this process is highly dependent on the type of protein. Also, protein crystals are very fragile, and the outcome of the process is really dependent on the operative conditions. Indeed, the proteins are sensitive to pH and temperature, and they can denature very easily. And this leads to a low probability of nucleation, and also you need a lot of protein to reach a very high supersaturation and induce nucleation, which means also very high cost. Nucleation is usually achieved by acting on the process parameters, such as the temperature and pH, but also ligands, polymeric precipitants, and salts, which act through the salting out effect, can be employed. And we can act on and induce nucleation through different methods. The traditional ones, which are microbatch, vapor diffusion, and free interface diffusion, which are commonly used, for example, for the screening. And more advanced methods, which rely on electromagnetic and ultrasonic fields, and on the convection-free crystallization, which of course is the topic of this presentation. So regarding convection-free crystallization, this is used to eliminate the convection and ensure transport through diffusion. This can lead to controlled supersaturation gradients and to a more uniform crystal growth. Particularly, this can be obtained through different approaches. The ideal one is microgravity, where experiments are performed in space, and this can lead to the avoiding of convection and an improved crystal growth and more uniform crystal structure, and a much really improved crystal quality, since you can reduce the incorporation of impurities during crystal growth. But this method is really costly, and it is logistically complex. Therefore, other methods have been proposed, such as microfluidics, which employs lab-on-a-chip devices where crystallization occurs in droplets. And so you can use small volumes, and so you have a reduced consumption of proteins and reduced cost. But this method is also difficult to, let's say, be employed, since it introduces the complexity of the device fabrication and the possible surface adsorption. So there is a third method, which employs gels, which can be based on silica or agarose. And in particular, gels, they represent a very attractive compromise, since they can reduce excellently convection at a lower cost. But it is possible that during this process, some gel-protein interaction occurs. And this is the main problem we focused in this work. So let's now dive more into details on gel crystallization. As I said, it eliminates convection and buoyancy effects, and it simulates microgravity conditions where homogeneous nucleation prevails, and we can reach a controlled crystal growth. And also secondary nucleation is almost fully inhibited. But not all gels behave the same. As I said, we can use agarose gels, which are commonly known as nucleation promoters. They have been, for example, introduced in a study from Artusio in 2021, when we saw that by increasing the concentration of agarose in the gel, there was an increased nucleation, so number of crystals, and these had lower sizes. These type of gels are called physical ones since they are formed by the entangled polymeric chains, while the silica gels are different type of gels which basically are nucleation inhibitors, as demonstrated by Gavira in 2013. These gels can act as inhibitors on nucleation since when the concentration of the gel was increased, there was a decreased nucleation of lysozyme. And also they differ from agarose gels since they are chemical ones, where the gel is constituted of cross-linked polymeric chains. So here comes the main question, which is why does silica inhibits nucleation and how can we control it? And to understand this phenomenon, we should first look at how silica gel forms. These kind of gels form through the hydrolysis of siloxane groups in tetramethoxysilane, which is used as a precursor of these gels, and the subsequent polymerization of silicic acid. And these gels, as I said, have a lot of advantages since they are able to create a stable environment where convection is reduced, but also they can unfavor sedimentation and provide very uniform crystals with a good structure, but also high mechanical properties, which makes these crystals very suitable for structural studies. And however, we can face different interactions between the gel and the protein, which is the reason for the inhibition of nucleation. And it is also the reason for the high need of protein concentration, and so the high cost related to this technology. So if we hypothesize if silanol groups are responsible for inhibition, can we chemically modify the gels to reduce this aspect and this effect? And so what we did as the core idea of this study was to modify these gels, which in the traditional way are only formed of TMOS, by including into the gels some additives which are methylated silanes, the so-called methyl diethoxysilane with 1 methyl groups, then dimethyl diethoxysilane with 2 methyl groups, and 3-methyl ethoxysilane with 3 methyl groups. So by progressively introducing these methyl groups, we aim to replace silanol groups with methyl ones and to increase the hydrophobicity of the gel to modify the network structure and also to reduce the protein absorption. And the idea was to make the gel more hydrophobic, but also to tune and assess the surface chemistry, cross-linking, and elasticity, so the gel properties. To do that, we employed an egg white lysozyme as a model protein, and we performed 2 different crystallization approaches. The first one is the counter-diffusion crystallization, where basically lysozyme and the precipitant, which is a salt, are separated from a gel septum, and the salt diffuses in the gel towards the protein. And the second approach instead is batch crystallization, which was here employed on silica gels for the first time. And in this case, in a PCR vial, we can add different solutions based on protein, of course, the buffer, then the TMOS, which creates the gel, and the precipitant, and they are progressively mixed to create an environment where the protein and the salts are in contact. Also, here we varied the concentration of TMOS used to obtain the gelation, and so the formation of gel. And also we introduced a new parameter, which is the additive to TMOS ratio, AT ratio, which was varied between 10 and 20 to also assess the effect of the concentration of the additive. And also we varied the protein and salt concentration to vary the supersaturation of the system. Here, I just would like to briefly outline the research project, which started from a counter-diffusion study where the concentration of TMOS and additives was changed to investigate the effect on protein crystallization. And then the same procedure was repeated in batch, where a higher control of nucleation could be achieved, since there are no supersaturation gradients, and we could better investigate the effect of the gel. And furthermore, we performed different release tests to evaluate the protein-gel interactions. And finally, we correlated the crystallization to the gel properties through rheology and contact angle measurements. So after this introduction, I would like to dive more into the actual results of the study. We started, as I said, from counter-diffusion crystallization, where we selected 5% of TMOS, since the 10% inhibited the nucleation. And also we employed, in this case, a constant additive to TMOS ratio, which was around 20%. And we selected a high protein concentration to induce nucleation of 100 milligrams per milliliter, and the concentration of salt, which was 200 milligrams to milliliters. Here we observed how the inclusion of the additives could modify the crystallization and the nucleation pattern of lysozyme. We could see that in this condition where 5% TMOS was employed, the additives could effectively modify the chemistry of these silica gels by replacing the silanol groups with methyl groups. And this resulted in an increased nucleation density as the number of methyl groups employed increased. And this also led to progressively smaller crystals, since we had a higher supersaturation and higher formation of nuclei, and so nucleation prevailing on crystal growth. And we could also see that the effect of methylation reached a sort of plateau. So after the inclusion of 3 methyl groups, there was a saturation effect, since no significant difference in size and number of crystals was appreciated between 2 and 3 methyl groups. So to better investigate this first screening effect of these additives, we moved to the batch crystallization. Here we compared the nucleation effect of 5 and 10% of TMOS for 2 different combinations of protein and salt, which were chosen to achieve overall a similar supersaturation inside the gel. So we compared the crystallization pattern at 5 and 10% TMOS for the two different conditions. And we saw that at 5% TMOS, we obtained overall smaller crystals, and the gel appeared to be more viscous, since the gelation was not so complete. And also, this looked like a sort of poorly of viscous gel, which may be useful for XFEL applications, but not for our application in this study. And also some parts of the gel appear to be collapsed, especially at the top of the vials. Instead, at 10% TMOS, we observed larger crystals, and also the gelation was complete, and this led to a gel which was stiffer, which formed within 1 day, and this allowed for nucleation to be subsequent to gelation, which is a good property. And this content, let's say, of TMOS appear to be better for growing crystal in batch for our application. The effect of the concentration of silica was also observed through scanning electron microscopy technique. So crystals were lyophilized, and then they were evaluated on their morphology was evaluated through SEM. And we observed that lysozyme crystals adapted to the silica-gel environment, and it incorporated silica fibers. Moreover, the concentration of TMOS did not affect the crystal shape, which appeared to be tetragonal, and this is coherent with literature, with the known form of lysozyme. So once we selected the optimal content of tetramethoxysilane, which was 10%, we investigated in the next part the effect of the additive to silica ratio. We selected a combination of protein and salt of 70 milligrams per milliliters of lysozyme and 40 milligrams per milliliters of salt. And the AT ratio was kept at 10 and 20%. So here we evaluated how the incorporation of additives altered the crystallization and so the nucleation. And we saw that basically the incorporation of additives increased nucleation and promoted it. It also reduced the amount of protein absorbed on the gel, and it decreased the crystal size due to the higher nucleation density. And this was coherent with the previous study in counter-diffusion. Here, the increase in the AT ratio provided a higher nucleation density for the first two methylated additives, while again a saturation effect emerged with the third additive. The saturation effect can be better observed if we look at the morphology of the crystals by scanning electron microscopy. So here we observed that it was really visible that as the nucleation density increased, we observed clusters of crystals, and the environment reached a plateau where the gel was saturated with protein crystal, and no more single crystals could be observed. So this is why we cannot, let's say, observe the significant differences between the 2 methyl groups additive and the 3 methyl groups 1. But overall, we then repeated this study for different concentrations of protein and salts, and we could assess that the control of lysozyme nucleation was obtained, and it was validated for a broader range of protein and salt concentrations. And so here we could see that effectively the control of nucleation could be achieved by modifying the gel structure. Then we wanted to better investigate which was the effect of the modification of the gel, and so the reason, the mechanism behind it. So we started by understanding the protein additive interaction in solution. And to do that, we performed a microbatch crystallization where we varied the protein to additive mass ratio. And we compared a ratio of 12, which was the same used in the gel, and a ratio of 0.4, where the concentration of additive was higher. In the first case, crystals maintained the tetragonal standard shape, and no alteration was observed compared to the additive-free solution. An increase in additive concentration instead led to tetragonal crystals, which displayed elongated 1,1,0 phases, which are typical of lower supersaturated environments. Indeed, in this condition, the precipitation of proteins which we observed may have reduced the concentration of free protein in solution, thus lowering the supersaturation. And this means that when the concentration of additive was the same of the gel, there was no interaction between the gel and the protein, since the crystal's shape was not altered. While when the concentration of additive was increased, there could be some interaction. So after that, we concluded that in gel-like conditions, the additive's action was mainly related to the modification of the gel respect to interaction of the additive and the protein. And so the next step was assessing the protein additives interaction in the gel. To this end, we performed release tests of protein from a gel to a buffer solution, which was placed in direct contact with the gel. Here in this graph, we can observe the release profiles in the presence of additives at 10%, the solid lines, and 20%, the dashed lines. And this was performed from different time points, from the first one, which was 24 hours, and then up to 140 hours. So here we could see that the highest recovery of lysozyme, and so the highest release of protein was reached through the modification with the 2-methyl groups additives, while the slowest release, and so the highest retain of protein in the gel, was found with the spare gel, meaning that there was the higher absorption of protein in the gel. And so the increase in protein recovery observed with the modified gel was related to the higher availability of lysozyme macromolecules, which were not adsorbed on the gel, which confirmed, let's say, our first theory. But here we observed an interesting phenomenon, and so the release of the gel formed with 1-methyl groups and the gel modified with 3-methyl groups were similar. And this is not coherent with our theory that with an increased number of methyl groups, there is an increased hydrophobicity of the gel. And so we tried to investigate better this aspect, and to do that we assessed the gel properties. So in the final parts, we started by contact angle measurements to evaluate the hydrophobicity of these gels. And we looked at the contact angle measurements, and we saw that the contact angle was the lowest for non-modified dry gels, indicating a strong hydrophilicity. while the hydrophobic nature of the gels increased with the methylation, but it did not scale linearly with the number of methyl groups, which is coherent with the phenomenon observed before, since we see here that the contact angle is not increasing anymore when 3 methyl groups are introduced. So to explain this, we also assess the, let's say, elasticity and the rheologic properties of the gel. We looked at the mechanical properties and we saw that the additives increased the elasticity and the cross-linking by increasing the storage modulus. The lowest storage modulus was observed for the non-modified gel. And this incorporation of additives could improve the elasticity and the extent of cross-linking. And we hypothesized that in the presence of TMOS alone, the resulting gel was less interconnected due to the scarce accessibility of residual silanol groups which are involved in the condensation reaction for the formation of the gel. And so when methylated additives instead are added to the system, the structure results in a more open one, where the accessibility of silanol groups is improved when the additives are present. However, the addition of 3 methyl groups results in a different structure where the storage modulus decreases. And this behavior was attributed to the excessive amount of methyl groups, which may have triggered the formation of local hydrophobic clusters and could have led to the reduced elasticity of the gel. So to conclude, we tried to modify the silica gels to control the nucleation and to overcome the inhibition effect of silica on protein nucleation. And we so could increase the nucleation density to with increasing the number of methylation of methylated groups into the gels. This led to a lower crystal size, which could also be tuned through the gel modification, but a saturation effect was observed when too many methyl groups were introduced in the gel. Also, we looked at the hydrophobicity of the gel, which increased up to the to methylation, and this was coherent with the theory that this methylation could reduce the interaction between protein and the gel. And it also increased the elasticity and cross-linking of the gel itself. However, we also demonstrated that nucleation is chemically tunable, but when we increase too much the number of methylation, we obtain excessive amount of methyl groups, local hydrophobic clusters, a reduced elasticity of the gel, and so the protein is more, let's say, absorbed on the gel than the 2 methyl groups. So let's say the nucleation control was valid up to 2 methyl groups, but these results could provide a rational pathway overall for designing convection-free crystallization media and to overcome the limits of silica gels. So with that, I am concluding. I thank the co-authors of this study. This is the publication which is related to the work I presented today. And I would like to acknowledge the co-authors, Roberto Pisano, Jose Gavira, and Fiora Artusio. So thank you for your attention, and I would be happy to take any question.
All right, thank you, Lorena, for the impressive research that you presented. Is there any question from the audience? I can break the ice if there's no questions with 2 curiosities more than questions. First of all, again, thank you for the presentation. First of all, do you think that the strategy you presented could be extended beyond protein crystallization to other fragile or highly sensitive molecular systems? And the second one is more closely related to my background. So I've seen an impressive experimental work and procedure that you established and successfully delivered, but I was wondering if there is or there could be any room for advanced modeling in terms of both first principle models and or black box machine learning model and or hybrid approaches.
Okay, yeah, thank you for the questions. Yeah, concern the first question, of course, this strategy poses a new basis for crystallization in convection-free environment, since this is a low-cost approach, which could be, let's say, more suitable for crystallization than microgravity approach, for example. This approach, I believe this could be tuned for other macromolecules. For example, I'm thinking about monoclonal antibodies or other poorly crystallizable macromolecules or molecules. So by inducing nucleation in a favorable environment, we could indeed promote the nucleation of other molecules. But of course, we should consider that crystallization is really dependent on the molecule chosen. And so this setup should be, let's say, adapted to the molecule of interest. And so some modification, of course, should be done. And concerning the second question, it's really an interesting one. Yes, this is an experimental work, but I believe that it could be possible and it could be really interesting to model this and to introduce a model to better understand the effect of these gels, both the inhibition and the promoter effect. So yeah, it's a great idea.
All right. So thank you again. If there are no other questions and also Boti does not have any, all right, we might proceed with the next speaker. So thank you, Lorena, again. I will share for the last time my second screen and present. All right, so next speaker is Professor Christopher Burcham from Department of Chemical and Biomolecular Engineering, University of Illinois, Urbana-Champaign, USA, and he will deliver a presentation entitled Ab initio Modeling in the Development of the Crystallization Processes for Pharmaceuticals. Again, also in this case, a brief background. So he's a professor of practice at Illinois since 2025, after serving as executive director in Lilly's synthetic molecule design and development organization. At Lilly, he led the particle engineering group and advanced crystallization and adsorption, enabling platforms for pharmaceutical development. His long-standing research focus is in separation science, especially crystallization and particle formation technologies. In 2024, he received the Ike Industrial Research and Development Institute Award. So thank you, Chris, for joining, also for you from all of us, and please, the floor is yours.
Antonello, thank you so much for that nice introduction, and thank you for allowing me to present today. Let me share my screen and get started. So, good afternoon, everyone. As was introduced, I'm going to talk about essentially examples that I think are a great opportunity in the pharmaceutical industry for the integration of digital design as it applies to solid state. Much of what Neda said in her great presentation earlier today serves as a really good backdrop for what I'm going to talk about. But to kind of frame the stage, I'm going to discuss the transition that occurs from a molecule coming out of discovery labs moving through development and ultimately into a medicinal product that you or I as a patient might take, and the importance of the solid state and the development of the solid state in that evolution of the molecule during that lifetime. And I'll point out that evolution starts very early in the development process. In fact, it precedes the development process because it actually starts in the discovery phase process. And so early on, when the probability of success for a molecule is quite low, we're still already investing quite heavily in the solid state. So if we look at the probability of success just from phase 1, forget about discovery, but from phase 1 clinical trials until we get to commercialization, at a very conservative, or very a level that is probably not very conservative, the probability of success is predicted to be about 12%. I think it's probably a bit lower than that, but we'll take that as a nice estimate. And it costs about $2.6 billion on average to develop a new drug. So there's a bit of a balance that the pharmaceutical industry undergoes with the development of every molecule, and that is this tension of not overdeveloping a drug versus underdeveloping it and then being caught on your heels at the time of registration or worse, at commercial manufacture. That timeline is anywhere from 10 to 15 years. There's examples of where it's much shorter, of course, but as molecular complexity is increasing, we see that this tension that exists between early development and investment versus the probability of success leads to opportunity. And that opportunity, I think, it really comes to how can we use digital design tools to decrease development efforts and ultimately the timeline of drug development. This question led to a publication that myself and a couple of colleagues from multiple institutions worked on, as shown here. And the question we asked is, what can we do with nothing more than a molecular structure to get to crystallization design? So to give a example of why I think this is important, I'm going to go back to a case study. And this is work I did many years ago when I was at Lilly and published on a couple of times. And we had a project that was for Alzheimer's and is in very late phase clinical trials, subsequently terminated. But as we were moving rapidly through phase 2 into phase 3, we had a problem statement where the dose was expected to be quite high, the order of 140 milligrams, and the tablet needed to be quite small for the geriatric population that it would be serving, so 400 to 450 milligrams. That leads to a drug product that would have a very large concentration of the drug substance. And typically, that's a challenge, but it's not an insurmountable challenge. But it is one that does require some effort. But in this particular case, the challenge was really quite excessive. The project started, like all solid-state projects start, with doing form screening. And you develop a phase relationship map similar to what's shown here, where we found that we had two anhydrous polymorphs enantiotropically related to one another, where that conversion temperature is about 45 degrees C, and also the formation of a dihydrate above 80% RH. There's a couple of unstable forms, but these were the three that were really in play. And as most, I think, companies would do, Form 1 was initially selected for development. And if that were the end of the story, there would be much of an example here. But in a particular case of Form 1, when it crystallized in extremely long needles. It was very difficult to even filter and get out of a reactor, let alone try to formulate. And so it was discovered very early on that Form 1, while thermodynamically the most stable form, was from a process perspective unprocessable, especially at the high drug loadings that were needed for this particular project. And so attention turned to the dihydrate form. And in fact, trials were started using the dihydrate. However, upon storage stability in the tablet form, it was found that the dihydrate And the dihydrate was formed because chosen because it had this really nice morphology. Fairly equine crystals, they flowed well. We could get to a high drug load. The tablets were very good compression strength. And so everything seemed to be great until we put the material on stability, and we found that in the formulated tablet, the dihydrate dehydrated, and it resulted in a mixture of Form 1 and Form 2. Not acceptable from a solid state perspective to carry forward, and certainly not very controlled. So it didn't fit with the idea of a quality by design product. So we started to turn our attention to Form 2. We turned it to Form 2 because Form 2 had much more equine crystals, but there was much concern and consternation about choosing Form 2 as the target form, knowing that at room temperature it was not the thermodynamically most stable form. We could get over some of that anxiety by looking at the conformation of the molecules in the crystal lattices. And so here I show the crystal lattice of Form 1. And if you squint hard at this, you can see that the molecule within the lattice is in a conformation where it somewhat wraps around on itself. And if you look at the conformation of Form 2 in the lattice, you see that it's much more planar. It also stacks one above the other, where in Form 1, you're seeing this sort of a head-to-tail stacking arrangement. This difference between the conformation of the molecule within the lattice and relative to one another within the lattice led us to postulate that the interconversion kinetics between Form 2 and Form 1 would be extremely slow. And as long as those interconversion kinetics were essentially non-existent over the intended shelf life of the product, then we knew we would have a kinetically stable polymorph that we could carry forward. So with that in mind, we developed a crystallization process, very typical cooling crystallization process, where we generated the crystals, we milled them to the desired particle size, we showed the kinetic stability that was needed, but we found after doing this that the initial product that was formed failed in the tablet manufacturing process at high speeds. And this was because there was a large number of fines, as you can see from this photomicrograph. The aspect ratio, while it was much better than in Form 1, was still undesirable. It was greater than a 5-to-1 aspect ratio, which from the literature would say you would have better flowing material. And something called a flow factor coefficient, a measure of the ability for the powder to flow, where large numbers are necessary, was low. We were finding flow factor coefficients of around 2, where it's desirable to be above 3.5. And all this resulted in a limit of our drug loading to less than 5%, which would have meant a tablet burden for a patient of about 6 to 8 tablets. This led to a very intense crystal engineering effort to improve flowability. We knew we needed to improve the crystal habit by decreasing the particle width and decreasing the aspect ratio as a result. We knew we had to reduce the fines. And so we did this through an experimental process of solvent screening, looking at the impact that the solvent would have on the growth of the different faces of the crystal. We looked at the solubility once we chose our solvent as a function of other solvents that we saw had a beneficial impact on our morphology, as well as an anti-solvent, which would help us ensure that we could control crystal form below that enantiotropic temperature. And then we developed the crystallization process utilizing slurry milling, temperature cycling, and solubility control. We resulted then, through this efforts, in a process that's depicted here, where this was a recrystallization and at the end of a crystallization at the end of a reaction. It came out of the reaction. We had to concentrate it through distillation, and then we found through these experiments that the addition of water helped inhibit the growth rate in the axial direction and promote the growth rate in our radial direction. And so we're able to increase the aspect ratio or decrease the aspect ratio of our crystals by the presence of small amounts of water in our solvent system. We seeded the crystallization and then we cooled it down. This was essentially the same approach that we had taken in the material that was generated previously but didn't perform well. At that point, we milled the material, and this was the change. We milled the product, and then we thermal cycled it. Why did we thermal cycle it? We did that so that we could dissolve the fines that were produced, as well as play on the difference between the growth rate and dissolution rate of the different phases. And in doing so, by the end of the third thermal cycle, end up with material that had a much improved crystal habit. All of this was done above that enantiotropic crossing temperature of 45°C. And by doing that, we were still in a thermodynamically stable region for that crystal. But we knew we needed to isolate it at room temperature. And so before we cooled it, we added an anti-solvent to greatly reduce our solubility. And then we cooled it to increase our yield. And then at the end, we milled it yet again so that we could get a much smaller aspect ratio. And in doing so, we went from crystals that looked, excuse me, very similar to what we had formed before we did the thermal cycling process to much more equant crystals. And you can see a great increase in the thickness of the crystals, as well as a decrease in the length of the crystals, and the aspect ratio being on the order of 3 to 5. So it was a great success. We were able to go from material that had poor aspect ratio and poor flowability to material that across a large number of scales had a much improved flowability. And when this work was initially done, we thought this was fantastic. This is a great model to carry forward. We're able to go from an impact a patient where they'd have a high tablet burden to one where it would have a much lower tablet burden through the particle engineering efforts. But this is a story that's an old story. It's one that's been told a number of times. We were able to increase the habit, like I showed. We were able to increase flowability. We're able to greatly reduce the RSD during tableting on the tablet weight. We're able to get a single tablet, and we're able to scale this from 500 milliliters up to 8,000 liters in scale and transfer it to commercial scale. So huge success. Why am I presenting this though? Because of the success, we've replicated this on many projects. But with that replication, many efficiencies have been gained. But here's the but. That still requires a lot of effort. There's still a lot of experimental work that needs to be done. And the workflow, while understood, is still laborious. And it gets to the point that Neda made in the rationale for why Takeda has made the investments that she showed in their digital workflow. Anything that can be done to reduce this effort is a gain. And one of those tools is crystal structure prediction. And using that to understand and maybe start to understand what form interconversion predictions could look like. Solubility predictions, as Neda showed, using various tools, and morphology predictions, and multidimensional population balances models once you have those morphology predictions. What if, though, instead of doing this during development, what if you did it at the end of the discovery phase, during that molecule selection, or what we call candidate selection in pharma? What if we integrated this into that effort such that the effort to develop the API could be estimated at the time of candidate selection? What value does that bring? The value is that it allows you to understand where you need to make your investments. Which compounds do you need to invest time in crystal engineering, as an example? Which compounds are going to be difficult from a crystal form landscape perspective? Which ones are going to be easy? Which ones can I hold off applying resources to until I have a better probability of success down that development pipeline? What if these considerations became part of the computational tool set that's actually used in discovery to select molecules to carry forward into experiments? So with that motivation in mind, let's look at some of these tools that are available. Crystal structure prediction is something that, if you look at the literature, is used more and more regularly. If you talk to individuals that are in the pharmaceutical industry, the ag chemical industry, you'll find that they're using these tools on a regular basis. And what they do is they really reduce the uncertainty during form screening and solid state selection. Solubility models, as we saw in the first presentation, broadly used in the industry. I think it's been widely adopted. However, some of the other tools are less so. So population balance models are not used regularly. They are used, but I think it's probably in some companies more on a case-by-case basis. And many times in some companies, you have to justify the use rather than expect the use of population balance models. Morphology modeling, sadly, I think even less so. So in all of these, in my experience, are discrete activities without interconnectivity. And this leads to the idea of ab initio modeling in the development of pharmaceuticals. What if you link these together and then ask the question, what would that process look like? How difficult would the process be to control the physical properties, the desired properties of my molecule, crystal form, crystal shape, and particle size? And so what would that linkage look like? This is what we came up with conceptually. You would start with nothing more than molecular structure, and you would run that through crystal structure prediction calculations, CSP0 calculations, and then refine that to bring that to ambient temperature through CSP-THD computations. What you end up then is a landscape that it looks at the different energies as a function of the density of your crystals. However, what we see, and what I'll show in the next slide, is that those that you see through CSP are not always what you experience through experimentation. And so we had a hypothesis that we need to understand primary nucleation, and we needed to be able to predict those homogeneous nucleation rates to determine, of those predicted low energy forms, which would be more likely to be observed experimentally. And once you had that, then you could start to develop the solid form landscape. You would understand what crystal form to carry forward. You could do morphology modeling and start to predict relative growth rates of the different phases in different solvents. You could do solubility modeling and end up then with the ability to do a multidimensional population balance model and get an idea at least of what that development process might look like and how easy or difficult it might be to control the desired solid state properties of your active ingredient. So this particular body of work looked at a number of different force fields to look at the crystal energy landscapes. And what we found is that the predictability of the CSP predictions is somewhat reliant on the force field that's used. And here it shows two different ones, and you can see experimentally forms 1, 2, 3, and 4 have been observed, and these are indicated by the colors, and these match up well with what's been predicted by the CSP calculations. And as you carry these through from CSP0 to the CSP-THD calculations at ambient conditions, you see that you get similar ranking between the two force fields, but you also see that there are structures that are predicted at lower energy that have not been observed experimentally. And for someone in the pharmaceutical industry, that gives you pause for concern, because that makes you ask the question, well, have we just not searched long enough experimentally to find those target forms? So this started to throw a kink in the question that we were asking, can we design a process simply from molecular structure when this well-studied example shows that while CSP predicts 2 additional forms, we're not seeing them experimentally? Why is that? And how could we better predict if you would see those from a kinetic perspective? So it's an open-ended question. Solubility predictions, as we've seen, are fantastic. Cosmo RS and other tools allow for predictive solubility from ab initio calculations. However, some data is required for them to be quite accurate. The heat of fusion is needed. some solubility data is needed or vapor pressure is needed. You need some experimental data to tie it back. However, we showed that this can be overcome through the utilization of molecular dynamics simulations to predict the solubility of one solvent. And then once you have that, you can then use that to then center your solubility predictions in Cosmo RS or other tools of that nature. Morphology predictions, actually we've had fantastic agreement between the experimentally predicted crystal structure, the crystal morphology is predicted from the experimental crystal structure and match up to experiments versus the morphology predicted just from CSP calculations alone for the two different molecules that were studied in this work. So, to summarize, these ab initio techniques have been matured to an extent that they're very routinely being used in the development of pharmaceuticals. However, they're being used in discrete instances. If we want to achieve the goal of complete ab initio design of crystallization processes, we found that the biggest gap exists, as one might expect, in the nucleation predictions, and especially with primary nucleation, because it's hypothesized that that's what's dictating which crystal form of those predicted by CSP is going to be the most likely to be observed experimentally. And then secondly, the ability to predict absolute solubility and absolute growth rate. So, how can we better get to growth and nucleation? I think one way is by understanding the role of molecular conformation has in solution on the growth and nucleation kinetics. And as an example of this, many years ago, I was working on a project that involved an atrope isomer. This is an isomer that has a rotatable bond and that it's sterically hindered. In this particular case, we were doing an anti-solvent crystallization, and we started with a seeded crystallization with a 20% relative supersaturation. And at this case, this is our typical starting point. The compound that should have very quickly desupersaturated in solution, and we should have seen crystal growth down to where we reached the solubility limit. However, nothing happened. There was no growth for days. And in fact, as we increased the supersaturation ratio to an excess of 2, we still saw no growth at all until we got to a critical anti-solvent level where we started to see growth. And it wasn't nucleation, it was just growth. And the question is, why did we see this change? And it was very repeatable. Once we got to that critical anti-solvent level, independent of our level of supersaturation, growth would happen. And what we determined is that it involved a conformational change of the molecule in solution. The addition of the anti-solvent changed the conformation of the molecule in solution and made it look like that of the lattice. And so we realized that there is something to be said for the impact of the conformation in solution to the conformation in the lattice and the energy that the molecule must overcome in order to make that conformational change. This led to some work that Peter's group did, where he looked at the theoretical impact that this interconversion equilibrium can have and found that growth impedance will become significant for such atrope isomers. Exactly the conditions where we saw where that steric hindrance can interfere with the ability of the solute molecule to change its conformation and to attach into the lattice. More recently, Cruz and Cabesa's group, along with Salvalaggio, have shown for a case where they looked at diacids of various carbon links at the conformational impact, and they found that the conformational change on the ability to crystallize was greatly impacted once the length of these linear molecules became to a critical value of around 7. And here you can see in this particular lattice, you have a nice linear orientation, but when you come in with one of these conformations, in order for this to dock into the lattice, the conformation of the molecule has to change. This is not a new observation. Going back over a decade ago, Derdor and others found the exact same phenomenon. They postulated the conformers in solution can have a long half-life and affect the barrier conformational change. So I think there's opportunity here to look at this on growth rates, but also the impact that it has on primary nucleation. Perhaps we need to think of nucleation as not a two-step process of a condensed phase into an ordered phase, but maybe it's a condensed phase into a conformational change into an ordered phase. And can this then explain why low energy forms predicted by CSP are not experimentally observed? Should we include conformational energy barriers in determining the lowest energy polymorphs that are most likely to nucleate? So with that, I'd like to thank the organizers and the audience, and I'd be happy to answer any questions you might have at this time.
Thank you, Chris, for this excellent presentation. I really enjoyed the fact that you brought out the problem that you could do the process level predictions from the molecular structure only, this ab initio workflow. And actually, many of the pieces are already in place, from the solubility, molecular dynamics, to the process level models. I really like that idea. Thank you for the talk.
Yeah.
Any questions from the audience? Until we have some questions, I really wanted to also ask this at the Neda talk, but perhaps there is enough overlap. Neda also mentioned that the project timelines in the industry are often tight. So how the in silico tools can actually accelerate the development, because these also need some time to develop, validate, and things like that. How this integrates into the practical time frame, if you could share your opinion on that.
Yeah, I think it comes down to that early I think it comes into two places, well, multiple places. But the first interface is that interface with discovery, where discovery is going through an enormous amount of computational chemistry to look for molecules that are going to dock into a target site to impact some disease state, right? It's the lock and key type model. From that, you'll get a large library of molecules that are then carried forward into different molecular computational tools to understand what the predicted efficacy might be, and then ultimately leads to a very large experimental effort in discovery labs to then synthesize those molecules. And then from that, from an efficacy perspective and toxicology perspective, then those molecules are carried forward into development. And it's only then that we really start to look at the impact of the solid state can have on the developability of that molecule. And by that time, you've made a very large investment in your discovery effort. And if you start down that path of development and into phase 1 clinical trials, you lead down a road that gets very expensive with time. And it becomes very resource intensive. And so I think there's a great opportunity that if we can better understand our form landscape, if we can better understand things like solubility, especially in vivo solubility, then we have the ability at that time when you're selecting molecules to carry forward into your experimental work and discovery to start to influence those selections. Right now, like I said, those selections are largely influenced by efficacy and toxicity. But what if we built in manufacturability? What if we built in expected in vivo performance? You would have better selection. I think you would have better selection. The other time is once you're in development, you have limited resources, and like I said, complexity is going up. So the time to understand your form landscape is growing because of that molecular complexity. And the number of targets that we're carrying forward is also growing. But the resources are the same resources. So how do you address these problems in a way that doesn't leave you on your heels down the road? You don't want another example like my case study. While it's great for a career, it's lousy from a development perspective, right? Because each one of those challenges means potential delay in the clinic, which means a potential delay in an important therapeutic to patient. So anything we can do to ensure that we do not delay the clinical progress of a molecule is a benefit, not just to the pharmaceutical company, but it's a benefit to patients as well. So I think there's a number of places where that can influence. the idea here is that you might start with digital design from a molecular structure, but it's not absent of data. So as you progress through the development, you've got these refined models. And then as Neda showed, once you start to have experimental data, you can further refine these models and continue to build them. The question is when do you start? And if we could start at that discovery interface or before, we could then really have a very good framework, in my opinion, such that when you get into development, you've got a good template to start with. And you also have a very good framework when you sit in a position like Neda does and has to say, where do I put my resources? This is where I want to make my investment. I'm looking. Do you want me to answer these questions in the chat, or will you ask me, Bolly?
Yes, I think we could answer one or two. So I think we have a question from Ivan Marciano, which is touching the problem of kinetics. He claims that we seems to be cracking the problem of thermodynamic solubility, but what about the kinetics? And he mentions that there was an excellent paper at Angevante a few years back, which used solution NMR to understand conformation in pre-nucleation clusters. So he's asking if that could be an important piece of the puzzle in the kinetics.
Yeah, so it's not a surprise that Ivan would ask this question. And maybe he's got some answers to crack this problem. But exactly, that's the tool that we use. We used solution NMR to understand that this change that we had in growth from essentially no growth to rapid growth was a conformational issue, and we discovered that through the application of solution NMR. And I do think that that is a tool that needs to be explored more and maybe utilized as part of that solid state workflow on a regular basis. Yeah. I think there's also computational tools that we can do to also evaluate that and what that steric hindrance is, what that energy barrier is from one conformation change to another. Yeah, excellent question. An excellent point.
Thanks. And what if we continue this kinetics and we try to put this into the PBM? I think Ashwin also had a very good question, which was that what do you think is the reason that PBMs and morphology models are not being routinely used despite there are several commercial tools for that?
I know,
Is it
and
a promiscualization mismatch or?
Yeah, I don't know, Boti. I think that's a great question. It's, there's Chrysiv, there's G-formulated products, there's other tools out there. People have developed their own, in-home models. I don't know why they're not used. It's a disappointment to me. We did utilize it in my experience. We found it to be extremely valuable, especially for continuous crystallization development or in problems like this where you're wanting to change morphology. I think part of it is the ability to easily predict growth rates of the different facets and then put that into a population balance model. That connectivity, in my experience, doesn't exist. And I think whenever you have to use multiple models and then connect them independently, it leads to another energy barrier. And this is a human energy barrier. And anytime you have an energy barrier, your kinetics slow. And so I think that's a big part of it. I know that efforts are underway at CCDC to better effectively allow for morphology models to be more democratized, and so maybe that will lead to more uptake in the community.
Yes, I know we are overdue with the time, but I really want you to answer just in very briefly about Christo's very intellectually challenging question. So this is about the conformations. So he was asking, should we screen more at higher temperatures, you believe, where conformations might interconvert faster? Could that give us a higher chance of getting the right conformations to observe certain forms?
So I think it's a fantastic question. I think yes. I think it's a great idea as long as we understand the relationship between different crystal forms. we want to ensure that we don't have some enantiotropic crossing point when we're at those higher temperatures and maybe fool ourselves into seeing stability that's more of an enantiotropic issue than it is thermodynamically stable at ambient temperatures. But I do think it's a great point. Yeah.
Thank you, Chris. Antonello,
And maybe if I could, I'm looking at that same question, and there's a point about bulk supersaturation doesn't mean much when you have the wrong conformation, and it's exactly the point, and that's exactly the point of the paper by Peters. It's not been picked up in the literature much, but he goes through a really nice derivation that shows that essentially, when we measure supersaturation, we measure just the concentration of the molecule. We do not measure the concentration of the right conformation. But in this paper, he shows the effect that conformation has. And if you have a strong equilibrium pushing you in the undesired conformation, it's essentially like you have zero supersaturation. And it totally explains mathematically why growth doesn't happen under conditions where you should see it. So yeah, so Christos's comment is a very valid one. Sorry, I didn't mean to interrupt you.
Yes. Okay. Yes, thank you very much, Chris. I think there may be left some unanswered questions, so I would kindly ask you to look at the Q&A area.
Yeah.
And I would like to thank you again for this very exciting presentation, and the depth of the questions also illustrate how important this topic actually was. And I would like to also thank to the rest of the presenters. I think it was a very nice and inspiring spotlight session. And I would like to give the word back to Antonello to add anything to the end. And yeah, thank you again.
Sure. So we are at the end. I would like to follow Boti's thanks to everyone. So first of all, thank you again, Chris, Lorena, and Neda for joining us as speakers today. Today's spotlight talks clearly showed both the breadth and depth of current research in crystallization, from control and digital design to in silico process development, protein crystallization, and ab initio approaches for pharmaceuticals. I would like to warmly thank again the EFCE Working Party on Crystallization and the organizers for making this event possible, and of course all the participants left and also who joined at the beginning for joining us today. We hope that this session has not only shared interesting scientific results, but also helped strengthen the connection within our community and stimulate further discussion and collaboration also. And thank you again. I do hope to see you all to the next EFCE organized event, and also to PBM 2028 in Torino. But if you don't have any other remark, I think we can close.
Yes, I just wanted to give the word back to Antonio if he wants to add some closing remarks to this session.
No, I don't have any closing remarks. Just to thank everybody for this wonderful webinar, and I hope to see you in the next EFCE event. All right.
All right.
Okay. Thanks
everybody. Bye-bye.
Thank you. Bye.
Thank you, everybody.